Hi, hi. My name is Kyron Savage and I'm doing biology at AS level. And today I'm going to be revising the biochemical tests for molecules. So, to start with, I've got my book here. We look at the Benedict's test for sugars. So, sugar is like a general term for monosaccharides and disaccharides. And sugars can either be classed as reducing or non-reducing sugars. And to test for these sugars, you can use the Benedict's test. And however, this test does differ on the type of sugar that you're testing for. So start off with a test for reducing sugars. And reducing sugars involve all monosaccharides. For example, glucose is a monosaccharide, and some disaccharides. Maltose is a disaccharide. And you add Benedict's reagent, which is blue, to a sample and heat it. So for reducing sugars, add Benedict's reagent to a sample and heat it. Make sure the solution doesn't boil. I don't think it will work if it boils, because if the test is positive, it will form a coloured, I can't say this word very well, precipitate, which is like solid particles that are suspended in the solution. And the colour of the precipitate changes from blue to green to yellow to orange to brick red. And it's really important that if they ask you in a test, you write brick red and say brick. If you write red, you won't get any marks. If you say orange red, you won't get any marks. You have to specify brick red. Apparently they use like crayons to colour code things in biology, ASL. So, and the higher the concentration of the reducing sugar, the further the colour change goes. You can use this to compare the amount of reducing sugar in different solutions, but a more accurate way would be to filter the solution and weigh the precipitate. So that's reducing sugars. But if you want to test for non-reducing sugar, like sucrose, first of all you have to break it down into a monosaccharide. And you do this by boiling the test solution with dilute hydrochloric acid and then neutralizing it with sodium hydrogen carbonate. So for non-reducing sugar, you boil the test solution with dilute hydrochloric acid and then neutralize it with sodium hydrocarbonate. Then just carry out the Benedict's test as you would for reducing sugar. But annoyingly, if the test is positive, the sugar could be reducing or non-reducing. So then you have to check if it's non-reducing, you need to do the check if it's non-reducing, you need to do the reducing sugar test to rule it out. To rule it out being a reducing sugar. So we've just done tests for reducing sugars and tests for non-reducing sugars. Next we have to know about is how to test for starch. And to test for starch, we use iodine. And this is a really easy one. You probably looked at this at GCSE or something. You've probably done this loads of times before. But what you do is you just add iodine dissolved in potassium iodine solution to the test sample. And if starch is present, it will change to a dark blue-black colour. Again, you have to use the words blue-black or they won't give you the mark. It can't be blue, it can't be black, it has to be blue-black. If there's no starch, it will just stay browny-orange. That's a really easy one. These are the last two. These ones I always forget. For proteins, we use the Biochemical test. And there are two stages to this test. The first one is that the test solution needs to be alkaline. So first you have to add a few drops of a sodium hydroxide solution. And then you add some copper sulfate solution. So the first stage is the test needs to be alkaline. So you first add a few drops of sodium hydroxide solution. So alkaline sodium hydroxide. Then second, you have to add some copper sulfate solution. And if there is a protein present, a purple layer will form. And if there's no protein, the solution will stay blue. But the colours are really pale, so you have to look carefully. So protein, purple, no protein, blue. I can't think of anything. Protein, purple, I can't think of anything for blue. Protein purple. I can't think of a word. Just try and remember. Just remember protein purple, and then you remember it for blue. So that's it. That's the Buret's test for proteins. So the last one is the emulsion test for lipids. First of all, you have to shake the test substance with ethanol for about a minute, then pour the solution into water. If the lipid is present, the solution will turn milky. The more lipid there is, the more noticeable the milky colour will be. If there's no lipid, the solution will stay clear. So that's a really easy one. So just think of lipid milk. I don't know. something relating to that. So let's go over them quickly. I won't say them all, I'll just say them what they are. Reducing sugar, use Benedict's reagent, heat it. If it's positive, it will form a coloured precipitate brick red. Non-reducing sugar, boil with dilute hydrochloric acid and neutralise it with sodium hydrogen carbonate. Then carry out the Benedict's test as you would for reducing sugar. The iodine test for starch, just add iodine, and if it's present, it will change to blue-black. Buret's test for protein, you need to make it alkaline, so you add a few drops of sodium hydroxide solution, then you add copper sulfate solution, and if there is protein, it will turn purple, and purple layer will form. For lipids, just mix it with ethanol for about a minute, and the more lipids present, the more milky it will be. So those are the biochemical tests for molecules. You really need to learn them, so just keep going over them. And good luck in your AS biology exam.